Good afternoon, students. Today we are going to talk about DNA as genetic material. So far we have seen what is gene, what is DNA, how come it is come. So now we have to talk about how the DNA is acting as a genetic material. So in till 1944 there was a discussion whether DNA or any other material protein was the genetic material. So 1940 they were thinking about protein was considered as a genetic material, as proteins are having the 20 different amino acids and they are in large quantity and they code for the more informations, and they had a confusions, and later on they decided no, not only protein is a genetic material, we also have some other thing that is nothing but a DNA is a polymer of 4 different deoxyribonucleotides, that is adenine, guanine, thymine, cytosine, and they are present in smaller quantities. So later on only they discussed and they proved that is DNA is the genetic material. So we have certain experiments which has been conducted time to time, and they only demonstrated that DNA only the thing which carries the genetic information and not the protein. So we have the evidences for DNA as a genetic material. We have 3 direct evidences which is proving the DNA as a genetic material. The first one is Frederick Griffith, which is in the year 1928. He was talking about the bacterial transformation and proved that DNA as a genetic material. Next is your Avery MacLeod MC Curtis method, that is also in the year 1944, and which is also in transformation. Then Alfred D. Hersey and Martha Case, 1952, he was having the experiment in bacteriophage. So coming for the DNA. So DNA is nothing but the deoxyribonucleic acid, which is recognized as the primary genetic material in all living organisms. And understanding of DNA comes, we have to know about what is the basic characters of DNA. So they have to know about the replication. The DNA can, they have to replicate itself. They have to increase their numbers during their cell divisions, and information, storage of information. So DNA contains all the informations which is needed for the development of the cell. So that is having the instructions for the development, functioning, growth, and reproduction of all known living organisms and many viruses. Then coming for the expression, though the process is like transcription, translation, the DNA will be code for the proteins which will be help for the functioning of the body. Coming for the transmission, so during reproduction, the DNA is being transferred from parent to offspring, and it is also ensuring the continuation of the genetic trait. So from one generation to another, it can be traveled. So this is the basic informations of DNA. So coming for the historical experiments, so we can talk about the experiments like Griffith, Avery, Hershey, and Watson and Crick model. So these are the models. So first one, Griffith model. So he was talking about the transformation in bacteria, suggesting the transforming principles carry the genetic information. And every method he was also talking about the same, which is a carrier of genetic material. And Hershey method, he concluded that DNA is a genetic material and not proteins. And Watson and Crick method is the double helix structure of DNA and how it is storing the genetic information and replicate. So coming for the experiments, so we have to identify DNA as a genetic material, very experiments. So first one we will be talking about the Griffith method. So he was undergoing about transformation. So he have taken the main object is to talk about the causing bacteria, pneumonia causing bacteria, Streptococcus pneumonia, and how it is having the processing. So coming for the method, so Griffith worked with the 2 different strains of bacteria. One is virulent, that is disease causing strain with the smooth outer coating, that is S strain, and other one is non-virulent, which is harmless, so strain without a coating, so R strain. So we have 2 different strain, S and R. So S is disease causing strain, R is non-virulent, that is harmless strain. So we are going for the experiment. So live S strain bacteria was injected into mice. So live S is nothing but it is having the disease. So it is injected into the mice. So once injected, the mice is dead. So coming for the live R strain, when it is injected into the mice, it is not harmless. So the mice is being survived. So coming for the heat killed S strain were injected, so that disease causing strain was heat killed. So it is having the things where it is this bacteria is not in a living condition. So the mice is survived. So when heat killed S strain bacteria were injected, the mice survived. However, the heat killed S strain were mixed with live or strain bacteria. So S strain and R strain both mixed. What happens? It is injected into the mice. The mice is died. So live S-strain bacteria was recovered from the dead mice. So what happens? Heat killed S-strain when it is mixed with R-strain. So they already dead, but when they have a living bacteria, they can imply into that bacteria and they can give their own replication process, and they are transforming their own things, and they got affected with that particular pneumonia, and the mice is dead. So from this we can conclude something from the dead S strain has been transformed to the harmless R strain into a virulent forming, and the Griffith termed this process. So, concluding, the dead S strain transformed the harmless R strain into a virulent form, and that is the process of transformation. So, it includes only the genetic information that is happens only of DNA. So, the next we are going to talk about the Frederick Griffith method. He was also so this is picturization of S3 strain and R3 strain. This is a picture where you can see the steps. So live S3, live R2, heat killed S strains. Heat killed S strains means live R strain. So this picture is explaining how it is steps. So the first one, the mouse injected with S3 strain, mouse died. Mouse injected with R2, mouse survived. Heat killed, mouse survived. So when the heat killed and R2, it is having the strain bacteria, and then mouse injected with heat killed S3 and living R2 strain plus DNA enzyme, so survived. So they proved that protease enzyme they are been died. So it is proved that no protein is been playing a role, only the DNA. So this is what the conclusions, and the Griffith is concluding that the material which is transformed helps is your DNA, not the proteins. So coming for the Avery method, Avery, MacLeod and M.C. Carty in 1944. This is also a main principle which was based on the transformation. So he was talking about the isolated different compounds, that is protein, RNA, DNA, and heat killed S strain and then separated with the R strain. So only the mixture containing the DNA from R strain caused a transformation. He proved that S strain caused a transformation while R strain into the virulent S strains. The enzyme used to destroy proteins, RNA and DNA. When DNA was destroyed, transformation did not occur, proving DNA was responsible for the transferring genetic information. So this is what the conclusion for transforming principles. So this is also been based on information, and we had RNAs, DNAs, protease. So in this we are concluding that the active factor and then transformation is only based on the DNA. In this they are talking about the DNAs and proteins, which is again having the confusions whether DNA or protein. So in that method he infected the bacteria, and these viruses are going to talk about the viruses which is having the labeling of this DNA of the bacteriophage with the radioactive phosphorus. So he labeled DNA with P32 and he labeled the protein with 35S, that is 35S. So he labeled both the DNA and the protein, and finally he come to know about what is happening. So the result was the radioactive phosphorus found inside the bacteria, indicating that DNA has entered the bacteria. The radioactive sulfur remained outside the bacteria. So sulfur is nothing but your protein code. It has not been entered, it is outside the bacteria, but DNA has been already entered, that is P32 is already entered in the bacteria. So this proves that DNA only the thing has the capacity to transfer. So this conclusion was being done and proved that DNA is a genetic material, not protein. So Watson and Crick method is also another one. He proved that DNA is double helix with the A, T, C, G and things, and he proved that it was been a genetic material. And coming for the Hershey and Chase method, and this Hershey and so in this we are talking about the bacteriophage experiment. So in this attachment of bacteriophage to receptor site on bacterial cell wall. So they have to attach, and then cell wall dissolves, and then phages DNA is entering into the bacterial cell, and protein coat remains out. So from this also they says DNA only the things which is entering into the cell, and protein which is present outside the cell. So this is what the method he proved. So this Hershey and Chase also concluded that 35S and 32S was the only thing which has been determining the protein coat and DNA coat, and finally they got the result. So phage is virus infect bacteria blender. Finally we got that is S in the supernatant and P in the bacterial pellet. So P is present inside the cell, and finally they choose that it is a process which has been held for the transformation. So this is what the labeled with P and 35S, and finally it concluded this P is being entering and not with the yes. So that is what the things which is conclusion, DNA as a genetic material. So these all the direct evidences. We also have some of the indirect evidences, that is equal amount of DNA is present in all cells of organisms we can prove, and DNA is in the presence of nucleus we can see, and divisions is happening only in the DNA we can see, and different species will vary with the amount of DNA. DNA is having the physical and chemical properties, and all the organisms have a great diversity of things, and ATGC is present, and this is one of the macromolecules, and it is main things. Some of the organism have, apart from DNA, RNA as a genetic material. In many of the viruses, RNA is playing a role, and they are the only genetic informations which is transferred from one cell to other cell. In case of all viruses, we can see sarcoma virus, influenza, hepatitis, and HIV, and all retroviruses. You can see about the transcription which is done for the processing. So coming for the plant RNA viruses, TMV, then Petunia, Cenedropen, papaya ring spot virus, potato leaf roll viruses, alpha mosaic viruses. These are some of the RNA which is having the genetic informations. So we can also talk about how RNA is a genetic material. So this is proved in tobacco mosaic viruses. And summary for the presentation, what we can say is expression is controlled through the central dogma, that is nothing but replication, transcription, and translation. So central dogma is should be maintained properly, and DNA is the only thing which is having the genetic information, and it is travel from one cell to other cell, not the protein. So this was the evidence for the DNA as a genetic material. Thank you.